So column chromatography is the topic for today's discussion. As we go to the principle and working or definition, surely it is about preparative mode of chromatographic separation. So we have stationary phase, we have mobile phase, as we had in the case of TLC or paper chromatography. And generally the stationary phase is silica gel, which is packed in the columns. The mobile phase, which is also going to be the eluent, it is going to be poured on the top of the column. It is going to run across the column, and it is going to move your analyte, right? So as mobile phase moves down, the compounds present in the mixture of the sample which is loaded on the column also is going to run down. So it is commonly used technique for most of the biomolecule separation and purification. So compounds are separated on the basis of polarity and the differential adsorption and of the analyte between the mobile phase and stationary phase. So depending on the polarity, molecules flow through the column at different rates, and separation occurs on the basis of rate of movement of these molecules through the different columns. Of course, we are going to apply high pressure to facilitate the movement of these analytes along with the mobile phase which is running down, but that is not the common technique. It is HPLC technique, high pressure liquid chromatography, where you apply pressure. If it is moderate pressure, it is MPCC. If it is high pressure, it is HPLC, like that it is talked about. But otherwise, with the help of gravity, the mobile phase runs down and the mixture is going to move down the column and get eluted from it. And eluted mobile phase is going to carry the analyte, rather I should say the separated analyte. And we are to collect the fractions of these analytes of these mobile phase aliquots and analyze those for the presence of the analyte, for which we have to use different methods and technique. So this is the schematic diagram to show how the column looks like. So generally, simple thing like your burette or pipette can be used to design a laboratory scale column, right? And it is filled with silica gel. And as you are seeing, here is the flask in which you have filled the solvent system. It is attached with your column. And of course, here is a filter disc kept onto which you are going to load the sample. So the filtered sample gets loaded onto the column. And as the liquid moves down, of course, it is controlled movement. You are going to regulate the flow of the liquid that is passing down the column. So here is a knob that shows you can control the rate. Of course, it is a very basic design that I am sharing with you. Automated machines for column chromatography are there. So, and here is a stopper one. Again, the rate of the eluent that is going to come out of the column can be collected at the precise rate. You can decide the volume per minute or volume per second flow rate out of the column that needs to be adjusted. Now, the stationary phases are typically alumina, silica, cellite, dextran, etc. What are they? We will discuss. But as in the previous lecture, we had talked about SPC columns, solid phase extraction columns. These are nothing but the preparatory columns where the sample of from biological source is going to contain a variety of components in it. It's a complex mixture, and the percentage of the analyte of interest may be less. So you have to remove all the unwanted interfering materials, and only the analyte of interest is to be concentrated. Say example, if you take urine sample or if you take plant extract sample, natural products. So you need to go for selective concentration of your analyte that can be achieved by solid phase extraction chromatography columns. This we have discussed in yesterday's lecture, so I'm not going to talk more on it, but if you want, you can refer to yesterday's lecture. Now, various stationary phase compositions are available. Ready to use columns are available on the market. When it is about the mobile phase, which is going to be added onto the top. You have to decide the adequate mobile phase composition and the flow rate so that you get differential migration of the analytes through the column and good separation is achieved. So this is the overview for column chromatography that we are talking about. So as I said earlier, you can have different materials which can be used to pack the column. So it can be silica gel, it can be alumina, or it can be polymer, synthetic polymers, which are cross-linked styrene, for example. And natural polymers also like destran, agarose, polyacrylamide, and even silica gel beads are commonly used. And for variety of purpose, column chromatography is applicable. Like for example, when it comes to separation of inorganic materials, porous silica, glass beads or hydroxyapatite is used. For synthetic organic polymers, when we say it could be polyacrylamide or polymethyl acrylate or polystyrene, and it is natural materials like polysaccharides. They can be used, which include cellulose, dextran, agarose, etc. So various matrix materials are available and are selectively used depending upon the analyte to be separated and the working of it. So let's see a few examples of these matrices. For example, this is a structure for cellulose. Cellulose is readily available in the nature and can be obtained in its purified form and can be used as the column packaging material. So as you can see in the slide, it is a linear polysaccharide. So we know what is the beta 1,4 linkage and how it is linked. So as you are seeing the structure of sugar molecule, that is beta D glucose, this is carbon first and this is the carbon fourth of the another ring of glucose. So it is beta 1,4 linkage between two glucose units, and polymerization is going to give us the cellulose units. Now, this cellulose unit can be put into bead forms. Of course, preparation of these beads can be facilitated by organic catalyst and reagents, and you can have the porous beads prepared from the cellulose powder. Now here I have attempted to share one photograph that shows the bead structure. It is the surface of the bead and this is the cross section of the bead. So you can see how the material porous is, and this porosity can be decided based on the quantity of cellulose used in preparing the bead gel, and that gel can be put into bead forms and beads are dried and are available. Of course, the columns which are packed with the beads ready to use, they are also available. So this is one example of that column, how it looks like. So biodegradability is one of the very important properties of this. Hydrophilicity, chirality, and of course it can be modified depending upon the requirement of the process. So you can add certain functional groups, selective groups onto it and modify the column depending upon the analyte to be used or separated. So the analyte will dictate what kind of modification of the cellulose bead is to be done so that adsorption on the column is better and elution would give you good amount of pure analyte. So one example of commercially available cellulose column matrix, it is a product name, of course, brand name, cellufifin. So here cellulose spherical beads are there and the particle size of the bead is 40 to 130 micron. So it is selectively prepared for separation of proteins and enzymes and biomolecules from their mixtures. Of course, as I said earlier, the cellulose bead can be modified or cellulose matrix can be modified. So here is an example ki the bead can be modified with lysine or polylysine or maybe with polyethylene imine so that it can remove endotoxins and DNA from biological samples. So if you have blood sample and if you want to retain specific DNA, then you can go with these kind of modifications. So selectivity is the essence of the bead that is going to selectively separate out the desired analyte. So this is overview of one of the matrix materials that is used in the column preparation, right? Some more examples are there. I'll talk about that and then we will talk about how the columns are are filled and all that. This is another example, that is agarose, which is 1,3 linked beta D galactopyranose and 1,4 linked 3,6 anhydro alphyl galactopyranose. So what is the repetition unit or repeating unit? It is galactose. And as you are seeing here in the structure, it is agarobiose is shown. So here it is 1,4 bond between the two is shown. So you have good properties like heat reversible gel is there, it is hydrophilicity is there, and of course it does not contain any other char groups other than the adsorption centers. So it is good as a matrix in the column chromatography. The hydroxyl groups which are available on these polymers, they can be activated. Activation is you can add selected functional groups or ligands and prepare it for variations of column chromatography. So when the variations of column chromatography we talk about, it is nothing but affinity chromatography, where you are going to add a selective ligand to it, or ion exchange chromatography, where a functional group, desired functional group is added so that uh, it can be converted into an ion exchanger resin or a cation exchanger resin, etc. So, to talk about the commercial products, Cifarose and Biogel A are made from agarose. Similarly, you have dextran, you have many more materials which can be taken as examples of matrices used. So cellulose, dextran, agarose, these are the natural ones. Similarly, you have polymer of acrylamide, which is cross-linked with NNN methylene bisacrylamide, and polyacrylamide, which is used for electrophoresis of proteins, for example, page gel that we talk about. So the same polymer can be used to fill the columns, and it would act as a matrix, and it is preferred because the stability is good. So pH range it can tolerate is 2 to 11, and same is the case with dextran. Up to very wide range of pH, these are functional. Even in case of agarose, 3 to 14 pH it is stable. So stability of the matrix, its inert nature, all these are very important properties when it has to be brought into practical application. So along with natural polymers, polyacrylamide gel, which is available in the market with the brand name, say Biogel P, is one of the examples. So can be used. Of course, silica that we talked in details in the previous lectures with respect to TLC also could be a good column material. So the silanol groups, SiOH groups, make it hydrophilic, and these OH groups are going to be the adsorption sites, and further it can be derivatized also. So excess SiOH groups can be removed. So for that you may give treatment with reagents, say trichloromethyl silane, and uh, the silica matrix can be further modified. Of course, again, the pH range is good, 3 to 8 stability is there. So it is one of the good matrix that is used when we are talking about the column, right? So is it clear? Of course, glass beads also is another common thing that can be used uh, as the column material. So when we talk about the columns, they are usually made up of glass and polyacryl plastic. That is the column itself in which we are going to put the gel bits, right? So generally the column length is about 2 to 70 millimeters, and length is about 15 to 150 centimeter. Length shorter than 15 centimeters, they are also available. It depends on the purpose for which we are applying the column chromatography system. So choice of the column dimensions is going to be dependent on the amount of sample that you want to analyze. If the sample volumes are large, you will require large column, right? But it is very important to remember that the column diameter to length ratio is one of the important things, and generally ratio of 1 is to 10 or 1 is to 100 uh, uh, these are the limits. That is, minimum it should be 1 is to 10, and maximum is generally 1 is to 100 diameter to length ratio of the column I'm talking. All right. So chromatographic column should be like a stable one, inert one. It is also provided with inlet and outlet. For loading the sample, you have inlet, and for elution, you have outlet. Um, then you have to attach certain adapters to it. So for eluting the sample from the column. So such arrangements are there. The standard column, how it looks like, that we have seen in the previous slide, but a detailed photograph we will share as we go with the actual working of the column. When it when it comes to, am I audible? So when it is to packaging of the column, you have to take into consideration that either you can pack the column with the slurry, or as we discussed in the previous slide, you can have the ready gel beads, right? That you are going to use as the matrix, and you have to fill the column with this slurry or the gel beads. So if it is slurry, then of course you have to prepare a thick slurry suspension. It should be degassed, so there should not be any air pockets remaining. Otherwise, it will affect separation of the analyte. So the slurry is prepared carefully, and it is gently poured onto the column. And the outlet part of it is to be like packed with gel, glass wool, so that the gel doesn't run down. Generally, three-fourths of the column is filled with the slurry, and the outlet is not open. So the column is, once filled, is kept for stabilization, and then it is washed with the mobile phase. So that is referred to as equilibrating the column. That is very required. So the buffer that you are going to use, or the mobile phase at a specific pH that you are going to use, it is run down the column. Just mobile phase alone, there is no sample loaded on it. It is just to equilibrate the column. For example, in case of TLC, we have seen to equilibrate the chamber, we prepare the solvent system in the glass chamber, and its lid is kept closed, and the vapor phase is allowed to occupy the void volume of the chamber. So the chamber gets saturated with the gas phase. Similarly, here you are going to equilibrate the column, so there are no gas space, or air spaces remaining, and a continuous smooth flow of the mobile phase takes place. So these are the some basic instructions when we talk about the column packaging and its activation or equilibration. Is it okay? समझता है?
Yes, ma'am.
Fine. So the packaging is completely done. It is allowed to settle, and then you are giving the wash with the mobile phase, right, to equilibrate the column. Then, of course, the ideal height and length diameter ratio, as I said earlier, it is not there, but the minimum and maximum sizes we have talked about. Now, when we talk about the column loading and application of sample, there are several methods, right? Before we go to that, let's go to the next slide. Right, the solvent systems that we have to talk in details. So I will skip this slide right now, and we'll go to next slide, where loading we have to talk about that of the sample. So you have to load your sample on the top of the column. So in previous lecture we talked about SPC, right, solid phase extraction columns. So that could be attached as a pre-column to your actual column. So separation of unwanted materials would take place, and the pure analyte of your desire can be chosen depending upon the type of SPE that you are selecting. It will allow all other materials to be retained, and only your analyte will be moving down that column or will be written at a specific distance. You will elute it out from there and then load your actual column. That can be done. All right. So when it comes to loading of the sample, as I said, it could be of two types. It could be wet loading, where you have the solution or suspension or liquid sample that is used, or it can be dry loading. Dry loading generally, for example, plant extracts, if you're talking about solid material. So when you're extracting it, if it is in the solvents, it's fine. But today, if I want to say analyze any of the Ayurvedic products that are on the market and I want to check what contents are there in it. So I'll have a dry powder, right? So I need to load the column with dry loading method. So generally in the dry loading, the sample is put into this solvent, is along with the silica gel, it is mixed. The silica gel and sample, generally the proportions are 1:1 to 1:3, and that resultant mixture is allowed to dry out again. So all the solvent is allowed to evaporate, and then that, with the help of spatula, you are going to load the stationary phase. So generally for natural product extraction, these dry loading methods are opted. All right. Now, there are, of course, several methods for application of the sample because it is to be applied onto the top of the column that you have prepared. So, very simple way is to remove most of the mobile phase from the column by suction and just to drain the remainder into the column bed. So the sample is then carefully applied. So you had to just remember that we had prepared the column, we had run the mobile face down. Now the upper portion of the column is going to hold some amount of mobile face. So try to suck out the mobile face a little a bit from that, and then apply the sample with the help of simply a pipette that you can do. So a small volume of mobile phase is then applied. So it is going to help regular distribution across the diameter of the column, and then the sample will run down the column bed. So more mobile phase is to be applied so that it runs, but it is applied to the column to a height of 2 to 5 centimeters, and then the column is connected to the mobile phase reservoir. If you see the previous simple design that we had shown, so this is your column, right? It is not fully packed. You see there is a gap, right? So here you are going to apply your sample analyte. This upper clean area that you're seeing is packed with the mobile phase, and then you are going to attach it to the reservoir. So these are the steps which you have to remember when it comes to loading of the column. Of course, there is an alternative procedures also are available, which include other manual modes as well as automated modes. So you have syringes which are used to load the column, and the syringe is attached to the syringe holder, and the syringe holder is attached to the chromatographic system itself, and it is controlled with the help of commands given or pre-designed commands, I would say, given to the system. Ki this much quantity at this much flow rate at this much height of the column is to be applied. So that injection of the column can be controlled by automated modes also. Uh, so there are different ways so that your sample is to be uniformly applied onto the column. Capillary tubing or a syringe or a peristaltic pump also can be used for loading. So mobile phase is fixed to the peristaltic pump. At a controlled rate, the mobile phase moves through the pump, and you are going to have different chambers which are going to hold the desired amount of your analyte mixture, and that is loaded onto the column. So of course, this is said to be most satisfactory method because a pre-decided volume of your analyte is mixed with the mobile phase. That is again pre-decided, prefixed volume of the mobile phase, and in a controlled manner, it is layered onto the column. And so it is most satisfactory and chosen method. So when it is about column chromatography, it is very important to remember what are its components, what are the stationary phases or matrices that are used, because many a times a short answer question also may be asked on such simple things which you may ignore. The examples are remembered, but what to write for 5 to 8 marks becomes a difficulty. So for that purpose, 2 3 slides I have put with the examples on the matrices used. Right, so loading of the column and illusion of the column also could be the topics that you will see in your syllabus. So coming back to the loading of the column or introduction of the sample onto the column, uh, you have to mix your sample analyte with something like, say, sucrose or ficol. Like, you remember in gel electrophoresis, while loading the sample into gel wells which are prepared in the gel, what we do, we mix it with something that would increase the density of the system, that is sample system. So you mix with sucrose. Right, so when you put your sample analyte into the well, it will not float away in the buffer, but it will sink at the base of the well in the agar gel. Right, so and in the similar manner, here you can add sucrose or you can say ficol to about, say, 1%, so that its sample density is increased. So the sample will sink below the top layer of the solvent. Otherwise, in the previous case, what we said, uniform mixing of some mobile phase and the sample you will do, and then it will be with the help of peristaltic pump it is applied. Another method is add ficol or sucrose about 1% quantity and then apply the sample. So what will happen, because the sample density is more, it will be settling at the uh, top of the column, but below the mobile phase layer. The 2 to 5 centimeter mobile phase layer is there. So in that, of course, 2 to 5 centimeter, I'm talking about a column which is 3/4 of the column size. Right. So based on the column size, you have to decide what will be the packed column size and what would be the mobile phase top layer dimensions would be. So here you are going to allow that sample to settle at the top of the column, but below the mobile phase layer. All right. So it is uniformly distributed on the surface of the column. That is very important. Otherwise, it will get staggered at one point where you are loading it and running down the column at that particular location. So separation will not be adequate and the entire area of the column will not be explored for better separation. So uniform loading is very important. Sometimes dyes like bromophenol blue, like in electrophoresis, also tracking dye and all that we use. Similarly, here bromophenol blue can be used in place of sucrose. So there are different ways of loading the sample, but generally it is with the help of syringe or capillary tubing, which is when you are doing it manually. Otherwise, with the help of auto control peristaltic pumps, you can load the sample. Right. So here in the next slide, you would see the schematic representation for how column chromatography would work. So in the first, there are 5 columns shown. In the first column, you are seeing the column is filled with the stationary phase, which is shown in blue color. The sample is loaded and above is the mobile phase to be poured onto it. So in the second column, you are seeing the mobile phase is poured on the top of it. Right? Then, as the time passes, the interactions between the solid matrix and that with the analyte would occur, and the analyte molecules will start running down the column. The movement is going to be dependent on the adsorption phenomena as well as diffusion phenomena, which are going to be governed by the characteristic properties of the analyte itself. So, as they move down, you will get separation of the analytes, analytes of the molecules of the same analyte will be moving at a particular rate, and the other analyte will either be moving ahead or remain behind In short, will get separated from the other analyte as interactions happen. If the interaction of the analyte with the column matrix is strong, it will be remaining on the column for a long time. It will not move fast. But if the interactions are weaker with the column matrix, then along with the mobile phase, it will run fast and elute first. So this was just an introductory talk about column chromatography. Are the basic concepts made clear to you? Any queries? Yes, fine. So overall, we have talked about what is the stationary phase, what are the matrices used, what is the design of a column chromatography column, what is the mobile phase, right? And how this column is prepared, how it is activated, how it is equilibrated, how you are going to load the sample and elute it. Of course, elution we have to talk in details with respect to the type of solvent system that is used. So mobile phase in details we have to talk. So within next